Hello everyone, welcome to the latest video in Omixon's Learn Me video series. My name is Alleles and I'm going to take you through this training video today. In this short video, I am going to guide you through the identification and investigation of novel alleles. Alleles that are not present in the IMGT database. Identifying novel alleles is straightforward in HLA twin. They are always distinguished by an icon next to each novel allele, a special naming convention containing the closest related IMGT allele, a hash mark, and an additional number to identify the actual sequence. We can start the investigation by opening the HLA typing sample results screen, selecting the genotype containing the novelty, and checking out the contents of the Show Novelties window. Here you can get detailed information including the gene region where it was found, the exact location within that region, and the type of the novelty. Types of novelty can be SNP as in single nucleotide polymorphism when one base has been substituted for another, insert when an insertion of DNA has been observed relative to the closest reference sequence, or delete when a deletion of DNA has been observed relative to the closest reference sequence. For further in-depth analysis, we will take a peek into the genome browser by clicking on the Jump to Novelty button. The novelty is highlighted by the locked cursor, and there is also an additional novel allele reference track and a related original reference track displaying the novel allele sequence and the closest IMGT allele sequence. We attempt to pick closest match for the IMGT database in terms of biochemical functionality, and we also use it to name the novel allele. In case of class 1 loci, we pick the allele that is the most similar in key exons 2 and 3, and for the class 2 loci, the priority is to find the closest allele on exon 2. It is important to remember that the name of a novel allele is not verified and just an indication of similar based on the sequence matching in the sample. In this example, if we have two class 1 alleles in the database where one has two mismatches on exon 1 and the other has only one mismatch on exon 3, then the former will be selected. This is important from the aspect of the expressed protein, and it also helps with the verification of the novelty. To check the validity of a novel allele, it is advised to add the base allele as a custom allele candidate and compare the alignments next to each other. If the Illumina reads map well to the novel allele reference and a coverage drop or gap can be seen on the alignment of the base allele, then the novelty passes this visual inspection quality check. If there are no Illumina reads supporting the novel variant and the base allele has an even coverage, then selecting the base allele as final genotype is suggested instead of accepting the novelty. Once you are certain about the presence of a novelty, please take steps towards submitting it to the IMGT database to help improve genotyping for the whole community. Omixon HLA twin software helps you with the first steps towards the independent validation of your new allele by exporting the consensus sequences straight from the genome browser. I hope you found this short video useful. If you have any questions, please contact us at support@omixon.com or place an inquiry with our sales team at sales@omixon.com. For more videos, please see the Omixon YouTube channel. See you next time!